Loud and clear.
Good. So I'm going to talk about blue-green algae. And as James just said, they're considered algae only because they have chlorophyll A and they conduct photosynthesis, just like a plant would, just like all the other algae. But again, they are truly bacteria. I'm going to talk about that. Next slide. So this is the families of organisms, everything from land plants, animals, and all the algae that we just heard about, and some we didn't hear about. And every colored box in this family tree of all life is a kind of algae. So cyanobacteria are down here on the bottom left. They are blue-green because of pigments. We'll talk about that. Then you have the euglenoids, the dinoflagellates. We know about red tide. We know about red drift algae, that red box in the rhodophytes down below. And then we heard a lot about some green algae, and there's many kinds of green algae. And some of the better ones for eating and feeding our food web are the diatoms and the chrysophytes. Those are the brown boxes up there. And those colors are really a reflection of the pigments. And to name an organism and classify it, they all evolved at different times on Earth. So we have to use pigments, but we can also use the cell wall. They have a cell wall like a plant does. Some are naked, but a lot of them have cell walls of different materials. Diatoms, for example, have a silica cell wall. Some of them are modal, can actually swim. Some actually just kind of oscillate back and forth, but that motility can help us figure out what things are. And of course, morphology, which is my specialty, understanding what these different forms look like under the microscope. And they all store different compounds. Some store starch, some store oils, all of which, again, is food for higher organisms. Next slide. So the classic organism that we worry about most is the Microcystis. So this is a close-up picture of Microcystis. I'm not going to call it aruginosa. It doesn't have the right morphology for that, but it probably is. And again, the key molecule that makes this cyanobacteria a bacteria is that insert over here on the right. That's a chlorophyll A molecule. There are other chlorophylls, by the way. There's chlorophyll B that we find in green algae and higher plants. But chlorophyll A is across the board in all of the algae. And just look at this colony for a little bit longer. Besides the big round organisms we see here, there's this array of little smaller rods. You see it in there. They're living in conjunction. They're actually part of the community. We call it the phycosphere. And they're an important function by providing nutrients to the cyanobacteria. Cyanobacteria aren't perfect users. These out there will take care of certain carbon compounds, sulfur compounds, and as they break them down, the cyanobacteria can use them. And that phycobiome that lives there is really important to understand. And some of my research is actually looking into that. Next slide. So again, the pigments are there that you might know about, the chlorophyll A. But what makes them blue is the phycocyanin. You hear the word cyan in there. And so some of the blue-green algae are quite bluish looking with a little bit of green mixed in. They also can contain another major pigment called phycoerythrin, like a red blood cell, erythrocyte. So you've got a red and a blue, and when they mix together, oftentimes blue-green algae look brownish, can even look black. So keep in mind when you're looking at a water body, they're not going to all be this blue-green color. So this array of pigments, what's it for? Well, it helps capture wavelengths of sunlight above and beyond what chlorophyll A can capture. So it picks up that full spectrum of light down into the reds and into the shorter wavelengths and makes them a very efficient organism at capturing sunlight. It's just part of their strategy of being so good at surviving in our water bodies. Next slide. So when these cells die, that's when sometimes you hear about turquoise. My water turned turquoise. It's the leaking of the pigments like phycocyanin here out into the water body. And it can look disgusting. All those little white spots are flies that are taking advantage of all the dead biomass there. But again, that's one of the first things you see when a bloom or organisms get trapped up on shore and they start to die. They don't want to lose the pigments. Those pigments don't have a need to leak from the cells. They only leak from cells when they die because it's a very big molecule to make. It's expensive. And it means something's wrong with the cells. They've run out of nutrients. They got sunburn, whatever it is, then you get leaking of that pigment out of the cells. Next slide. So one of the things I'm trying to do in my lab is to really understand what these organisms are. We know Microcystis. That's pretty obvious. Although if you take a deeper dive into the genetics, there's hundreds of strains of Microcystis. Some are toxin producers, some are not. And if you don't have the genes, and that's what I'm showing on the right by showing you DNA, you'll never make toxin if you don't have the genes for making it. So there's a lot of other organisms in our water body. Lake Okeechobee is full of many, many organisms that people didn't know about, because we've done a full genetic sweep of the organisms in Lake Okeechobee and then down into the Caloosahatchee. So by looking closely at the morphology, and that's what that picture is on the right, that green arrow is pointing to a specialized cell called an aconite. It's a resting stage. Microcystis would never do it. But we have another big bloom former in our lake and in Caloosahatchee. Right now, there's a ton of, it used to be called cylindral spermopsis blooming in the Caloosahatchee. And that other cell on the right is called a heterocyte. It used to be called a heterocyst. That cell takes N2, nitrogen, out of the air and fixes it. And I'll talk about more about that later. So again, I like to take images like this, but then I isolate these organisms. And with that, I can find it, do metagenomic sequencing and figure out, do you really have the genes for making toxins? Because a lot of times we put up these alerts in general because the water has got blue-greens in it, but we don't know if they're toxin producers. Next slide. They are really, really good at doing one thing that keeps them up at the water surface. They have a specialized structure inside the cells. It's called a gas vesicle. It's not a vacuole like you would hear with a higher organism because there are no membrane-bound organelles because it's a bacteria. But you see how big that structure is? That fills with air. It helps them bob up to the top of the water where they can maximize photosynthesis. And that capture of sunlight is really important to make them successful. So once they are up there, they're making basically a sugar compound, that C6H12O6. It's actually glycogen, a lot like what we make. And they make big, long chains of it. That's what the N is. And as they do that, that molecule gets bigger and bigger inside the cell. It collapses these gas vesicles and it causes them to sink. So that's part of their strategy. They come up first thing in the morning and they produce a lot of photosynthate and collapse the gas vesicles and they sink back down. This can take a day, it can take a few days, but whatever it is, once they sink back down to the bottom, you know about nutrient regeneration from sediments, especially the ones that are low in oxygen, like phosphorus comes out. Well, while they're down there, they might as well pick up a load of nutrients so that they can move back up. So this organism on the left is a Dilocospermum, used to be Anabina. Microcystis also has this mechanism, and not all genera have it. Some that are on the bottom don't have gas vesicles, but a lot of them do. It makes them very successful. Next slide. So why is this important? So if you look at water bodies that are used for drinking water, if you have the option to take water for drinking from a different place, that's great. But the cyanobacteria, what they'll do is that initial distribution might be even throughout the lake. Buoyancy causes them, like I just showed you, to come up to the top. And then wind, they're so easily moved by the wind. That's why when we go out to sample, one day they're right in front of us, the next day they're on the other side of the water body. So sampling is a little difficult for these organisms. But if you're a drinking water plant and you have your options, you can take water where there is not a bloom. And that's, in Florida, we have some surface water intake systems and they have to be careful. because of what's coming up next. Next slide. Another aspect of cyanobacteria, because they're bacteria, they're thermophiles, which means they love warm water. 25 degrees centigrade and above, fine, 30, fine, 35 even, 35 degrees centigrade, which is really warm water, they thrive. So if we look at this map that NOAA generated in 2018, the bloom that came down to us, you can see June 12th, not much there. June 20th, okay, there's a fairly big patch in the southern part of the lake. 4 days later, look how large that patch is. And June 29th, it's covering most of the lake. That all may not be growth. It could also be organisms that decided to float up because they were in the water column. But it just shows you the huge problems. This is Lake Okeechobee. That's a 30 by 33 mile body of water, and it has a couple outlets. A lot of it comes our way. Next slide. So one of the reasons we care a lot is the human health aspect of this. These organisms make toxins, and these are the major groupings of the toxins. The hepatotoxins, which means it affects the liver. It basically slows down the functioning of the liver. It can act more or less slowly, sometimes overnight, but days to weeks. And all the microcystins fall into this category. And there's another compound called nodularin, looks very similar structurally to microcystin. And then the cylindrospermopsin. Those three compounds fall under the hepatotoxin group. Okay, so yeah, it affects the liver. And but it's still quite deadly if you get too big a dose. The next group down is even worse. They're the neurotoxins. You have anatoxin A, you have guanotoxin, used to be called guanotoxin AS. You have saxitoxin and neosaxitoxin, and you might recognize those words from paralytic shellfish poisoning. It's the same compound. It's actually the same pathway that probably evolved first in the cyanobacteria, and they passed it on to the dinoflagellates. Then you have toxins that just, hurt your skin. So they're called dermatotoxins. They produce rashes. And, some people are very sensitive to this. And then the last compound that I'm going to talk about is BMAA. It's potentially, and I underscore potentially, linked to ALS and other neurodegenerative diseases like Alzheimer's and Parkinson's. But it's still potential because there's a lot of work to be done on that. Next slide. So how potent are these? So the most potent, well, first of all, lethal dose where 50% of the animals you inject on a microgram of toxin per kilogram of body weight of the organism is parts per billion. Look at saxitoxin. It's way up there on the top of the chart on the left. Okay, racine, which comes from castor beans, is more potent, but saxitoxin is more potent than cobratoxin. Anatoxin, AS, well, I should have called it guanotoxin, is as potent as cobratoxin. Microcystin, the one that we care about the most because it's the most common, at 50 micrograms, it's less potent than cobra, but it's more potent than curare and strychnine. And you get my point. Actually thinking about this, the exposure route, how do you get these compounds into you, is very important. These studies are all based on ingestion or direct dosing, where you inject a mouse. You can't get people to volunteer to do this study. So little has been done on understanding inhalation. And in my lab, we are spending time working on inhalation. Next. So these are little videos. If you could play one of them. Yep, there we go. So the inhalation study, what we're doing, and you can play the one on the right, is we are taking a known amount of algae from the water, from a bloom, or from culture where we know what the toxin profile is, and we're forcing it to make aerosols. And those aerosols, we tried just using wind over the top, and we really didn't create them. A little more difficult than what happens with red tide. But by doing a tight quantification of this, we hope to say how much is getting into the air, and therefore how much into, let's go to the next slide. how much might be getting into our lungs. So this device on the right, I took the concept from Mike Parsons. It's called an air impactor. It's meant to mimic a lung to some extent. It's basically layers that air can flow over. The top layer would be more like our trachea, and the bottom layer is more like our alveoli, the sacs at the bottom of our lung. And we look at the algae and the toxins that flow out of this chamber, and we analyze the different layers. And this is funded by Florida Department of Health. And again, we've looked at cultures, and we've looked at natural blooms to try to understand how much is coming out. Those data are forthcoming. We're not ready to share them, but I will share one thing. We're finding entire organisms are getting blown out of the water. It's not just the toxins. So when those organisms come out, they do have toxins in them. So we're rethinking how people are addressing this, and it's not just gases that are coming out. Next slide. So the toxins that are regulated, well, I shouldn't say that. The toxins where there's guidelines from EPA are microcystis and cyanobacteria. There's drinking water guidelines. They were promulgated in 2015 after the event in Lake Erie. You all know about the drinking water situation that happened up there. And it's 0.3 micrograms per liter of microcystis and 0.7 for cyanobacteria. There's some caveats. That was meant for children younger than school age and exposure for 10 days or longer. So it's coming into your water supply. They let older people be exposed to a higher dose. So again, these are guidelines. I think your bigger drinking water plants are monitoring for these compounds, and they will take appropriate actions to prevent these compounds from getting out and being in their distribution system. After that, in 2019, they promulgated recreational guidelines for all age groups. No 10 days. This is if you fall in the water, if you have 8 micrograms per liter of microcystins or 15 of cyanobacteria, those are the criteria for that. And that's why you see a lot of warnings put up by Florida Department of Health before they may even know the concentration. If there's cyanobacteria out there that they recognize, they will post in an abundance of caution. So these are good guidelines. It's meant for, people that are kayaking, people that are boating that might fall in the water, kids on the side of a water body. Hopefully there's no alligators. But that's what the recreational guidelines were based on. They're based on that children, how much they might engulf. And they got it from swimming pools. Next slide. So another piece of work that we're doing is under the innovative preventive technology, part of FDEP funding, is in the Pahokee Marina, which most of you know. And earlier last year in April, May, it was a total mess. We didn't get in there with our technology trial until September. So here's the marina on the left. It's about 9 acres. And the concept of this is using nanobubbles. So this little picture here, diagram, shows you that the bubble sizes that people talk about. And nanobubbles are supposed to be so small that they are neutrally buoyant. They're not going to bubble up to the surface. When this machine is on, there are macro bubbles that come up. We're not talking about those. These micro bubbles are supposed to be able to attach to cells and burst. And that bursting, we're saying, might have an effect and actually rupture the cells. So we're doing that. We're analyzing it. We're going out every 2 weeks in the Pahokee Marina. We're looking at the nutrient content. We're counting the number of algal cells. We're measuring the toxins to see if this technology works. Next slide. All right, to wrap up, keep in mind that when you go to a water body, the lifeguard isn't there, bacteriologist isn't there, the microbiologist, nor the phycologist. That's what I consider myself. And so swim at your own risk. And remember, you can go to the next slide. Cyanobacteria are actually beautiful. Here's a microscopic image of a known bloom former. It's called gliotrichia. Those red filaments in there are full of chlorophyll. That's why they're red. This organism has been boiled and it didn't die. And all the green is a DNA stain, and all the bacteria that live on this colony are lighting up. It just goes to show you that there's a lot to know about cyanobacteria. Thank you.
Dr. Rosen, thank you for the presentation. We have time for a few questions.
I've got a question. Dr. Rosen, this is Raymond Iglesias, Hendry County Commissioner and also General Manager at Roland Martin's Marina. When you speak about the Pahokee Marina, is the exacerbated algae that was in that marina caused by Microcystis and Lake Okeechobee, or more so from the boats that were docked there dumping fecal matter into the marina basin?
No, I don't believe so. There was a huge bloom that got caught in there in September, and very little activity from the boats, but that bloom came right in off the lake. It spiked all of our measurements. We were way above. And a couple of days later, it washed out because they've taken sheet piling out, and it flows a lot better. There's circulators in there now, and of course, our control mechanism. So I don't believe that would be it, because then you'd have a constant bloom. It wouldn't be this, oh, there's one now and it's gone. So I don't think that it is that nutrient source. The lake's got plenty of nutrients in it.
Okay, so the reason I ask is I know that their pump out system was not in operation and there was a bunch of liveaboard boats. So that's one thing that we don't allow at the marina. Is it's just reality is that someone that's on a liveaboard boat really doesn't want to go the extra mile or a couple hundred yards to move to a pump out system. So they do what's easy and they just, flush right in the water. No one's going to see it. And the reality is, in my opinion, the way the marina is designed, like he's talking about, the nutrients do blow into this marina basin and they can't get out until they move that sheet piling. So you're a doctor, I'm not, so I'm going to go with what you say.
Well, and I agree that, and maybe last April, May, when it was so bad, that's before they removed the sheet piling. And there could have been a lot of sources of nutrients built up from the sediments coming in, and of course the organisms being inoculated from the lake. At any given time, the wind blows the right direction, you're going to get a huge pulse of organisms into there.
Right. Also, with the wind direction of how it blows in that part of the county or the lake, the nutrients that are embedded into the sediment in that basin, I think, get reintroduced to the water column right in that area. That also.
Yep.
I've got a question. Bill Veach from Fort Myers Beach. You mentioned that these that in their cycle they get nutrients from the sediments. Is that their primary source of nutrients? Because I know we talk a lot about nutrients in the water, and a lot of way we get the nutrients out of the water is to allow it to settle. And I'm wondering if that's maybe we need to be more focused on the nutrients in the sediment as well as the nutrients in the water.
So the worst thing for cyanobacteria is have flowing water. So the only way they can move up and down and harness from, release from the sediments is if the water column is somewhat stable. So no, that primarily they want to get it from the water column. Microcystis needs nitrogen to make the toxin microcystin. Dilocospermum, another common bloom former, makes its own nitrogen from the air. So if that's off the table and we're just looking at phosphorus, if it's coming in from the watershed, if it's suspended from the sediments, it's got to be pretty much dissolved for them to get it. They do have some enzymes to take in organic phosphorus, but they really want inorganic phosphorus. And one, I didn't mention this, but when they run out of a nutrient, a key element, they can get stranded at the top of that water body, which is why we have guacamole. They lost their ability to move up and down. A key element ran out in their environment and they got stranded at the top. So after a big biomass accumulated. So.
And so you think it's the excessive biomass that starves them whenever they need to?
Oh, absolutely. That excessive biomass causes the fish kills. It's not the toxicity. It dissolved oxygen goes way down, and if it's a big expanse, the fish can't get away from it. So you have this huge sag and dissolved oxygen at nighttime when they aren't photosynthesizing, and they're just the bacteria is in there chewing things up and respiring. And then, the stuff that leaks out of the dead cells, which feeds more bacteria, causes more dissolved oxygen issues.
But the question is does it build up a certain amount of biomass that kills the bacteria itself?
No, it's just a symptom. So that buildup of biomass, that guacamole, is unhealthy cells that are dying and leaking and causing heterotrophic bacteria to grow and proliferate.
And just one more quick question. I don't mean to dominate here, but when you talk about doing the micro bubblers, the aerators,
Yep.
If there are macro bubbles in there, does that create an aerosol? Is that taking some of these toxins and putting them in the air?
Well, it could be. I mean, yeah, anytime you're aerating, you have the potential to cause aerosolization. The concentrations we're seeing so far are so minute, and there are no human health guidelines on those minute concentrations, especially from that exposure route of being in the air. But I'd imagine that there is some of that going on.
Thank you.
I have a question. When you were talking about the micro bubbles,
Nanobubbles, they're really
Yeah.
small.
smaller bubbles, not nanobubbles, bursting and potentially causing the release of the toxin if it's damaging the cell wall. Is that happening throughout the water column as well? Or you mentioned that those bubbles don't float to the top. So is that kind of through the cycle of going up and down? It could happen at any depth?
So these bubbles are supposed to be distributed evenly throughout the water body. And what they're actually doing to the cells is kind of an unknown. So FDEP is funding a new round of study. And as a side project, we're actually going to do this in the laboratory where I can really take a peek, look under the microscope, look for physiological indicators of cells rupturing, look for release of toxins, because it's theory right now. They're selling a lot of these units. They sell them a lot for agriculture, horticulture, things like that, aquaculture. But they're controlling algae. Are they working? We don't know that yet.
Thank you. Dr. Rosen, again, thank you for joining us today. Thank you for the